These are actually two different species. Mussels are hard to photograph. There's a federally threatened species of mussel that's known to historically be found in these lake systems, in Gantt Lake and Point A Lake as well. These being hydropower dams, we know about the aging infrastructure of the U.S. These dams are going to need routine maintenance from time to time, and some of that being more extensive. So for this project, they're actually having to replace certain structures on the dam, and it caused them to have to draw down the entire reservoir down to river level. Knowing that this federally threatened species was here, Power South got involved with U.S. Fish and Wildlife, having conversations about, what their proposed plans were and the knowledge of this federally threatened mussel and kind of what do we need to do at this point. So that kind of initiated with Point A Lake, and this is basically kind of a replica of what happened there, but on a bigger, better scale.
Oh, that's a Megalonias nervosa. Hold on, that's the shell. That's the other part of the shell.
Okay.
Yeah.
So that's just a piece of it, but this is actually, the common name is washboard. And they found one alive in the lake, and then these guys found
genus and species of that?
No, the remainder of this part of the dead shell. The live one they found, though, I believe, was about that big. It was huge. I mean, it was as big as a dinner plate.
Megalonias nervosa.
This one's called washboard for the common name, and if you had the whole shell, you can see these, they're called plications, these big ripples going down the shell, and it kind of looks like the old style, scrub your clothes on it washboard.
I started this project again, previously in fall 2017 with Mr. Miller as our principal investigator. Now we're repeating the same study but with better methodology, better techniques, but also much more organized. We flagged all the mussels rather than getting zeros previously.
Whenever they draw down these reservoirs, they basically put a large effort towards trying to find any of these federally listed species, and in addition to several state listed species that are in here, that they'd be able to go out and search for those and then relocate them back to a safe place where they can survive. Basically came out and surveyed the lake bed and moved a lot of these federally threatened mussels. There was, I think, over 250 found in Point A Lake, which was the largest known population back in 2017. And then in this lake, I think there were over 3,800 mussels of that federally threatened Fusconaia escambia. It's called the narrow pigtoe, I believe, that we were able to find in this lake. We took measurement data on all of those, put individualized tags on them so that future research can be done. So now each one of these mussels is uniquely marked with its own number. Now that's an old dead shell of the federally listed one there. And that's called Fusconaia escambia. Basically, what tends to happen is those that become rare or threatened or endangered are kind of a specialist as far as their niche is the word we use, where they fit in a habitat or in the ecosystem. Those that are specialists, if you change something that organism requires specifically, they just start disappearing. Those that are generalists tend to be able to live in all types of habitats and changes they can live with. So one of the major things that's been impacting mussels in Alabama and even in South Alabama is just merely dirt, sedimentation, erosion. Over the past, we've had so much erosion and sediments go into the streams and rivers that they've literally changed. And you have critters that live on the bottom. You have all your insects, your macroinvertebrates. There's tons of larvae. You have worms, you have crayfish, you have mussels, you have certain species of fish that stay on the bottom. But whenever you completely change that bottom, then you basically blanket all that heavy. Those species don't have anywhere to go anymore. They wanted somebody to come in that had a lot of expertise in mussels and kind of study what impacts it have on the mussels, which you can generally think, well, they're going to die, but how long can they live? What conditions are more favorable for them to survive throughout time? What species are more likely to live? What age classes maybe do better than others? And that's a lot of the research that we're doing, is trying to tease apart some of those different things. But you have a lot of different species and a lot of different characteristics of the species. So these being so thin-shelled here, these all died within just a day, a week of this lake draining. But these are common. We call them like a weed species. They're just everywhere. This one's a common one, but we're actually still finding these alive.
So basically, I'm measuring the temperature and the moisture and the millivolts of the quadrat that I am gonna dig, and determine the depth and the cover. And later when we dig the quadrat, and we're gonna determine the substrate, either it's clay or rock or silt or whatever we're gonna find, or maybe even detritus or some leaflet at the bottom. We don't know. We'll see.
The neat thing is you come over here and you see something like that and you say, oh, I think that was alive. But you can't pick it up and find out if it's alive. Because then I'm manipulating it inside of that unique habitat. And the idea is that we leave them there until we actually excavate that site. The neat thing is whenever we first started this study in Point A was U.S. Fish and Wildlife and myself and Power South, we all had these communications about the project, and we said, well, it's going to be 13 weeks. How long are they going to live? Is there even any point in planning this 13-week study? Because I think 14 for Point A. Because we all kind of said, well, by week 3, week 4, especially week 6, somewhere in there, everything's going to be dead. We're going to be wasting our time out here digging around. And, and what we found 2 years ago was, no, in the right conditions, that mussels can live at least 14 weeks out of the water. This looks like a really nice shell, and it is a really nice shell. But I can look at it and tell that this one actually died before the lake was drained. One of the differences being the internal shell color here. So whenever they're really fresh dead, they're kind of a pearly, purplish color. We have the typical labs, where students get out and they do a little bit of their own research depending on the course we're talking about, but it's still kind of the view, the mentality of, it's still a class. You kind of get that same kind of perspective to where when we get these opportunities to actually come out and do research, and you get to drag students out in the field with you that have the motivation, that actually want to get out here and do this stuff, they tend to develop various skills that you just don't have the time and the details to cover in some of the labs. So these guys come out, and it may not, or it may have directly to do with their future jobs, but just all the kind of relationships between the ecosystem and the different species, and kind of what is the key factors of allowing these organisms to survive, and not just the kind of experiences that they take so much to move into the career field, but the experiences that they take from this just as an experience, the pure enjoyment.